Good afternoon, everybody. My name is John Gallen. I'm director of the Clinical Center, and I want to welcome everybody to the 14th Annual Astute Clinician Lecture. This lecture is part of the NIH Director's Wednesday afternoon lecture series, and I bring my warmest greetings from Francis Collins, who regrets that he could not be here today due to some other pressing issues. The Astute Clinician Lectureship was established in 1998 through a gift from the late Robert W. Miller and his wife Haruko. Mrs. Miller regrets that she's not able to join us today. But I am honored to acknowledge the role that she and her late husband played in establishing this lectureship, and you can read about his role in the pamphlet that was distributed. The series honors a U.S. scientist who has observed an unusual clinical occurrence and by investigating it has opened an important new avenue of research. And certainly our speaker today is an excellent example of such a researcher. Dr. Kenneth Fishbeck, who goes by the name Kurt, is an NIH Distinguished Investigator and Chief of the Neurogenetics Branch at the National Institute of Neurologic Diseases and Stroke. He's a graduate of Harvard and earned his MD degree at the Johns Hopkins School of Medicine. He then went on and interned at Case Western Reserve University, completed a neurology residency at the University of California in San Francisco, and then went on for a research fellowship at the University of Pennsylvania. In 1982, he joined the neurology department there, serving as a professor from 1995 to 1998. He came to the NIH and his current NINDS position in 1998. Kurt is a fellow of the American Academy of Neurology and was elected to the Institute of Medicine of the National Academies in 2000. His laboratory is studying the mechanisms of hereditary neurological and neuromuscular disorders, particularly the polyglutamine expansion neurodegenerative diseases. Please join me in welcoming Dr. Fishbeck, who will speak on developing treatment for hereditary neuromuscular disease. Kurt?
I'm really pleased to be here with those of you who are here and for the home viewing audience we have by the webcam. And it's a real honor, kind of humbling for me to be recognized as an astute clinician. Let me just, before I go much further, give some disclosures. As an NIH employee, I don't take money for outside activities, but I do serve as an unpaid advisor, as a member of advisory boards for several organizations, including nonprofit voluntary organizations like the Muscular Dystrophy Association, the Spinal Muscular Atrophy Foundation, a couple of European nonprofit organizations, the French Muscular Dystrophy Association and a European Union consortium, and a couple of companies, Biogen, IDEC, and Prosensa, and I'll be mentioning Prosensa later in this talk. Although I don't take money from these organizations, I certainly am sympathetic with their goal of reducing the burden of the diseases that I'll be talking about today. So maybe this is something to keep in mind as I go through my talk. Now, when I think of an astute clinician, having spent time as a student in Baltimore and on the faculty in Philadelphia, the person who really comes to mind as the model of an astute clinician is William Osler. And in the time I was at Penn, my office looked out on the site of the old Philadelphia General Hospital, a Civil War era hospital where William Osler saw patients, which was torn down and replaced by a complex of medical research buildings, including the Abramson building. And in the lobby of that building is a saying, carved in stone, is this saying from William Osler, which I think is a good one to fit with the talk I'm giving here, is to wrest from nature the secrets which have perplexed philosophers in all ages, to track to their source the causes of disease. These are our ambitions. Well, over the last 25 years now, we've been able to fulfill Osler's ambitions for those diseases which are hereditary, Mendelian diseases that have genetic causes. Through the fruits of the Human Genome Project and all the work that's gone on through the Genome Institute here, we have tools at our disposal which allow us to map and identify disease genes as never before. This is a process that's mapped out here in a kind of cyclical way that starts and ends with patients. The first step in this cycle over here is to see the patients and characterize the disease manifestations, the phenotype, and to collect samples, to use these samples, DNA samples from blood or saliva, to map and identify the disease gene. This has been done now with several thousand human diseases, several hundred neurologic diseases, and each one is a great discovery, really. Solving a kind of experiment of nature, figuring out how specific lesions in the genome cause specific manifestations in patients. And it gives us a very useful, reliable diagnostic tests. But it starts us back on the lower part of the curve, because with the disease gene in hand, we can start to figure out how is it that the mutations affect the normal function of the gene? What is the normal function of the gene? Do the mutations cause a loss or gain of that function, a new toxic activity? We can recreate the disease manifestations in cell culture and animal models. And we can use these cell culture and animal models to work out the disease mechanism and to develop assays that can be used to test potential treatments. That could be small molecule treatments or biological treatments, gene correction, gene replacement, or small molecules that mitigate the disease manifestations. We can test these treatments in the animal models and select from those treatments, those agents which look to be safe and effective, to take them into the clinic in early phase and then later phase clinical trials, with the goal of eventually coming back to the patients with a safe and effective treatment. Now, we've made enormous progress around this cycle in identifying disease genes and working out disease mechanisms. The step which remains to be closed for the diseases I'm going to talk about, Duchenne muscular dystrophy and spinal muscular atrophy, is this last step. We have treatments now that work very well in animal models. If we were taking care of mice as patients, we'd be quite well off. But we have to be able to convert that treatment, that effective treatment in animals, to effective treatment in patients. There's a lot of work still to be done, but we're getting close. We can really see that goal. It's almost in sight. We're into the clinic now with specific treatment, rational treatment that's based on the disease gene. This is where the challenge remains, to get back to patients with treatment that's been proven to be safe and effective. So I'm going to go through that story now with two diseases that we've been involved in for a long time: Duchenne muscular dystrophy and spinal muscular atrophy. Here's a picture of a patient with Duchenne muscular dystrophy I saw years ago at Children's Hospital in Philadelphia. Actually, a patient that I followed from first diagnosis at age 3 or 4 through his death in his 20s, through the whole course of the disease. Patients with Duchenne are usually normal at birth, start to become weak at around that age of 3 or 4. The weakness is progressive. It affects primarily the proximal muscles at first, the hip and the shoulder muscles. And as the disease progresses, it affects all of the voluntary muscles, the respiratory muscles, and there's also cardiomyopathy. The patients typically become wheelchair bound around age 10 or 12 and survive usually into their early 20s or mid to late 20s nowadays, and then die from progressive disability, difficulty feeding, and difficulty breathing. The pathology that underlies this disease is a degeneration of the muscle. We see on muscle biopsy here in a picture taken from Alan Pestronk at the Washington University in St. Louis, a typical image of muscle fiber degeneration. The normal muscle fibers in cross-section look like these around the periphery, but in the middle are muscle fibers that are breaking down. You see necrotic fibers with vacuoles and an influx of inflammatory cells that gobble up the debris. Now, muscle has a remarkable regenerative capacity, and we see on the right here regenerating fibers, small fibers with basophilic cytoplasm that are proliferating and fusing to form new muscle fibers. The muscle goes through repeated cycles of degeneration and regeneration, but eventually the regenerative capacity of the muscle is depleted and the muscle becomes replaced by fat and connective tissue. The patients become weaker and eventually succumb to the disease. Well, 25 years ago now, in 1986, the gene for Duchenne muscular dystrophy was identified. This was one of the first disease genes to be identified by positional cloning. It's an X-linked recessive disease, and the gene was found on the X chromosome. It's the gene for the protein dystrophin, a muscle protein, and the mutations in this gene cause a loss of the protein dystrophin in the muscle. The dystrophin gene is one of the largest that's known. It's nearly 2.5 megabases, takes up 1% of the whole X chromosome, 0.1% of all of our DNA, contains 79 coding exons, some of which are widely dispersed with non-coding DNA in between. Becker muscular dystrophy is a milder disease that's caused by mutations in the same gene, but mutations that have less severe effects on dystrophin cause a reduction in dystrophin without a complete absence, and the disease manifestations are correspondingly less severe. Now, dystrophin is known now to be part of a structural complex that, well, it underlies the muscle plasma membrane and connects to sarcolemmal or plasma membrane proteins in the muscle plasma membrane that in turn connect up to the basal lamina on the outside of the cell. So dystrophin forms an important link in this structural complex which holds the muscle plasma membrane together as the muscle fiber contracts and relaxes. In the absence of dystrophin, this link between the internal cytoskeleton and the external matrix, the cytoskeleton or the structural proteins on the outside of the cell, the loss of this link causes the muscle plasma membrane to be unstable. And this is what leads to the muscle fiber degeneration that occurs in the disease. So dystrophin is an important structural protein in the muscle plasma membrane. Mutations in these other members of the complex, the membrane proteins that form the complex, and the proteins that form a link with the basal lamina, mutations in these other proteins lead to other forms of muscular dystrophy, limb-girdle muscular dystrophy and congenital muscular dystrophy. So it's an important complex for muscle integrity. Now, most Duchenne dystrophy mutations are deletions, internal deletions, deletions of part of the gene, one or several of the exons, and the deletions are not distributed uniformly across the gene. Here's an early study that was done in Leiden in the Netherlands showing where mutations are located. And we see here from one end of the gene to the other, the 5' to the 3' end of the gene, that there is a cluster near the 5' end of the gene and a cluster in the middle of the gene. These may be parts of the gene that are particularly prone to mutation or particularly prone to mutations that show up with the phenotype of Duchenne muscular dystrophy. Now, the Duchenne deletions take out an odd number of nucleotides such that they shift the translational reading frame. And the result is that the messenger RNA, the 3' end of the messenger RNA, the message is lost. And this part of the message, the transcript from this part of the gene, encodes an important part of the protein that's responsible for its linkage to the muscle plasma membrane complex. And a loss of this complex means that the protein, if it's just truncated, is unstable and rapidly degraded. Now, so this C-terminal part of the protein is important for protein stability and function. The Becker mutations typically do not shift the translational reading frame. They lead to a protein product which is shorter but still functional in Becker dystrophy. So we know now that the dystrophin gene defect leads to instability of the muscle plasma membrane and muscle degeneration. There is a compensatory process of muscle regeneration which eventually gets depleted, and this leads to the progressive muscle weakness of the disease. So with this knowledge in hand, how do we approach treatment for this progressive and ultimately fatal disease? Well, you know, one approach is to replace the defective gene. A challenge because the gene is so large here. but there are truncated versions of it which could well work. Or to correct the gene defect, usually at the messenger RNA level, and I'll be discussing each of these in turn. Another approach is to block the deleterious effects of the gene defect, to slow, perhaps with protease inhibitors, the process of muscle degeneration, or to enhance muscle regeneration. And I'll say a little bit about each of these approaches. First, in terms, working downstream in approaches that increase the regeneration of muscle, something that would be useful not only for Duchenne and Becker muscular dystrophy, but for any muscle disease, perhaps even including the age-related muscle weakness that we all get as we get older, so-called sarcopenia. Somebody's nodding in the back there. We all get weak as we get older, and a treatment that promotes muscle regeneration or slows muscle degeneration could be beneficial to all of us. But muscle, it's now known that muscle growth and regeneration are enhanced by specific factors. Insulin-like growth factor 1 is a factor which stimulates muscle regeneration, as is follistatin. And correspondingly, there's an endogenous factor called myostatin, which blocks muscle regeneration. That's also something that we need to prevent our muscles from overgrowing. Increasing IGF-1 and follistatin or decreasing myostatin have beneficial effects in a mouse model of Duchenne dystrophy. So years ago, a mouse strain was identified called the MDX, or muscular dystrophy X-linked mouse. It's been available as a disease model. These mice are less severely affected than patients with Duchenne dystrophy, but it's a very good and faithful, otherwise faithful animal model for what happens with muscle, to muscle with an absence of a loss of dystrophin. And myostatin inhibition and IGF-1 and follistatin overexpression have beneficial effects in these mice. Now, myostatin, as I mentioned, is a factor that suppresses muscle growth, and animals have been identified. A series of animals, dogs and cattle, have been identified that have mutations in myostatin, and even humans. One family has been reported with a mutation in myostatin that leads to an overgrowth of muscle, but this loss of myostatin is something that's well tolerated in this family. This is a report from Germany in New England Journal several years ago showing a baby with a complete loss of myostatin who has overgrowth of his muscle. So the myostatin inhibition, which is well tolerated in humans, increases muscle mass and strength in these MDX mice. Based on these findings, Wyeth, a major pharmaceutical company, undertook a multi-center trial of myostatin antibody, monoclonal antibody myostatin, in adult patients, so the more mildly affected muscular dystrophy patients, and did not show a benefit in terms of change in muscle strength. This is a study of over 100 patients. But Wyeth, the same group at Wyeth and a biotech company, Acceleron, have been working since then to develop safe and potentially more effective, more potent agents to work on this pathway, the myostatin pathway. So there's still a possibility for treatment to emerge from this. Another approach which has been pursued, well, by us and others over the years, is to use drugs that correct the translation of mutant messenger RNA. So it's been known for a while that there are drugs that will promote the read-through of premature stop mutations. So these are not deletions, but nonsense mutations that lead to a premature stop signal. These drugs, commonly used aminoglycosides like genomycin, are known to have this effect. And it's kind of like you see here, not so much on campus, but out in Bethesda, that when the cars get to stop signs, they just keep going. These are drugs that induce translational infidelity, that induce a misread of the stop signal, and promote a read-through that leads to full-length protein. Now, a company in New Jersey, PTC Therapeutics, developed a compound, specifically developed a compound much more potent than the aminoglycosides at inducing translational read-through, and they found that this was reported in Nature a few years ago. This drug was remarkably effective in MDX mice. So here are control mice. Here you're staining for, they're staining for dystrophin around each muscle fiber. You see the little green circles around each muscle fiber. MDX without treatment has very little staining. An occasional fiber with a somatic mutation that corrects the read-through, the stop signal. But then with the treatment, very nice expression of dystrophin in skeletal muscle, the leg muscle, the tibialis anterior, the diaphragm, and less so, but some expression in the heart. So again, based on these findings in the mice, PTC entered into a multi-center clinical trial, and again, unfortunately, did not see an effect on the primary endpoint of that trial, the 6-minute walk test. Now, there is some effect at lower doses. They looked at 2 doses. The higher dose did not show an effect. The lower dose did, but it's overall not a significant confirmation of efficacy of this approach to treatment, but further studies are ongoing. Interestingly, this approach of promoting read-through of mutations that lead to a premature stop signal is something that could be applied and is being developed by PTC as an approach to treatment to other hereditary diseases with this kind of mutation. So what about more biological approaches to treatment? What about gene replacement? As I said, replacing the whole gene is difficult in Duchenne muscular dystrophy because the gene is so large. There were attempts early on in the mouse model back in the early 90s, not long after the gene was discovered, using adenovirus to deliver the gene. Adenovirus has a large capacity, and it led to some delivery of the gene in a functional way such that low levels of dystrophin could be expressed in the muscle. But as we've come to learn subsequently, adenovirus is remarkably immunogenic. Our immune system is well designed and developed to deal with adenovirus and to attack cells that are expressing adenovirus. So it's not very safe for delivery in humans. What has been shown as a kind of proof of concept, largely through work done at the University of Washington in Seattle, Jeff Chamberlain and his colleagues, has shown that if you take truncated versions of the dystrophin, so these are smaller versions of dystrophin that can fit in other safer viral packages, and overexpress them in the MDX mice, so genetically correct, you can genetically correct the disease with overexpression of truncated dystrophin. And they went on to use adeno-associated virus, a safer, less immunogenic virus, and showed that they could deliver truncated dystrophin with intravenous delivery in the MDX mice. Here shown again the MDX mouse without treatment and with treatment. You see with treatment a nice ring of dystrophin staining by immunohistochemistry around each muscle fiber. Unfortunately, when they tried to scale this up to dogs, there's also a dog model for Duchenne muscular dystrophy, they got an enormous inflammatory response. So there's something about the dystrophin itself or about the AAV delivery that led to a real problem with inflammation in these dogs, and that slowed down the adaptation of this approach to humans. Subsequently, the group in Seattle was able to show that immunosuppression was something that could counter this inflammatory response, but immunosuppression, heavy immunosuppression together with viral delivery is something that is a little risky. Nonetheless, there have been human trials which have shown some indications of efficacy. First, there were an early version of cellular therapy, myoblast transfer, that was done in the 90s, usually gathering cells from an unaffected family member, injecting the cells into the muscle. Could see that that would replace dystrophin to some level, but usually at a very low level in the muscle. And then an experiment that was done with direct injection of the DNA into the muscle. A group in France here published a paper with injection of the gene into the muscle, biopsying the muscle afterwards. Here's a control muscle. Here's a muscular dystrophy patient muscle. After the injection, you see a single muscle fiber here. They could show that this was a a fiber that was positive in expressing the dystrophin that had come from the injection, but there were very few of them. I like to point out that this paper, which had 20 authors, had fewer than 20 muscle fibers that were positive. But the AAV, so the AAV constructs that were developed by the group in Seattle initially have now been used in clinical trials. The issues in these trials are first safety. You don't want to have the kind of thing happen to patients that happened to the dogs with the early trials in the dogs. The immune response, which is a big factor in the safety, the immune response to the vector, the viral vector, or the gene product. And then the delivery, efficiency, stability of expression, and the dose required. So the dose required for a human is a lot more than the dose required for a mouse, and then the production capacity to be able to make that much AAV. But a group in Ohio has done a good job in moving this field forward, I think. Jerry Mendel and others at Ohio State University, now Nationwide Children's Hospital, used AAV first to deliver a gene that is defective in limb-girdle dystrophy. So a gene for one of those proteins in the muscle plasma membrane that dystrophin binds to, alpha-sarcoglycan. They looked at 3 patients, injected into a foot muscle, the extensor digitorum brevis muscle. They found in biopsying the muscle afterwards, here the untreated side and here's the treated side, you see very little expression without treatment, and then with treatment, again, those rings around each fiber, which is very nice. They did see some antibodies that developed in these patients. There was 4 to 5-fold increase in expression of the protein, but in the presence of antibodies. The group in Ohio went on, this study was reported last year in the New England Journal, to use AAV to deliver this truncated or mini dystrophin to the biceps muscle of 6 patients with Duchenne dystrophy. And again, they saw very few positive fibers. And maybe a little bit concerning, they saw circulating T cells that were specific to dystrophin. So an indication of an immune response and low level of correction. Now, what's been, well, particularly exciting here the last couple of years, the last few years, is another approach to treatment, another approach to correcting the genetic defect in this disease with antisense oligonucleotides. So this approach, which is called exon skipping, is based on the finding, again, that Duchenne muscular dystrophy is usually caused by gene deletions that shift the translational reading frame. So antisense oligonucleotides can be designed that will promote skipping of the exon that contains the mutation or exons downstream in such a way as to restore the reading frame. And again, lead to a protein product that's a little bit shorter than normal, but can be quite functional. So it's kind of diagrammed here. You have exon, exon, exon. You have a mutation in this exon that perhaps leads to a stop signal, or a shift in the reading frame that leads to aberrant amino acids and then a stop signal. If you have an oligonucleotide that targets the splice site of this exon, you can induce the splicing machinery to skip over that exon in such a way as to restore the reading frame downstream. Now, it's nice in theory. It had been an idea for a long time. What has allowed it to get into, well, to get ready for prime time, I guess, is that chemical modifications of oligos have been developed that way enhance their stability, so that oligonucleotides that are injected now can last for weeks and months and have a long-lasting effect. Now, this has been shown very nicely in the MDX mouse. Again, nice correction. Here's untreated mouse muscle, and mouse muscle has been treated with oligonucleotide to skip over the mutation in the MDX mouse. And it has been shown in patients. So first in cell culture experiments, this is a group at Leiden University at the time, showed that a oligonucleotide targeting exon 51 first leads to skipping. This is the messenger RNA amplified. It shows that the transcript that you get in untreated cells and the transcript you get in treated cells, and you see the skipping of the exon leads to a shorter messenger RNA produced. And here's a muscle cell in cell culture that's been treated with this oligonucleotide, and you see nice levels of expression here in longitudinal view of this muscle fiber. And several years ago now, this group reported in the New England Journal treatment of, let's see, 4 patients with Duchenne muscular dystrophy with injection into the muscle. And very nice expression of dystrophin in the muscle after direct intramuscular injection. Here are the levels of expression. It's between, 17 and 35% of normal levels. Here's normal muscle, Duchenne muscle. Here's muscle, Duchenne muscle has been treated by this approach with injection right into the muscle. Another group in England and the UK using a different kind of oligonucleotide showed a very similar effect in results that they published a couple of years later. So 2 different groups, now 2 different companies involved in developing these and bringing them into larger scale clinical trials. The group in Leiden, together with a biotech company called Prosensa, and later a large pharmaceutical company, GSK, has now done a multi-center trial with subcutaneous delivery. Remarkably, injection of these oligonucleotides, not into the muscle, but under the skin, leads to systemic correction of dystrophin deficiency. It's here biopsying the patients after 7 weeks after subcutaneous injection, subcutaneous treatment. You can see expression levels that are approximately, or up to about 10% of normal. So it's not fully correcting the disease by any means, but the idea is that maybe given enough time that the expression levels will be much higher. Now remarkably, in this study, it was really just 2 sites in Europe. 10 patients participated in the study, have been followed now for, well, they reported in this spring for 1 year, now up to 2 years, and 7 of the 10 have had an improvement in 6-minute timed walk. Now, there's a lot of scatter here. This is not a significant effect. You see here untreated patients, not a randomized control group, but contemporaneous controls. Patients without treatment tend to have a decrease in timed walk. They'll have some improvement up until age 8 or 10, and then they'll crash. They'll become very much unable to walk, no matter how much time it takes. But the patients who were treated with the oligonucleotide, well, 7 of the 10 of them had improvement. Now, you can look and see for yourself whether this looks like a difference. It's clearly not a significant difference. But it's a remarkable effect in some of these patients who had had this kind of treatment. Now, based on this study from Prosensa, GSK has entered into a major development effort that included first a phase 1 dose escalation study in Columbus, Ohio, and then a phase 2 study to look at different doses in Europe and Australia at 12 sites. And then a large scale for a relatively rare disease such as this, a very large scale phase 3 study, a pivotal study that's being done worldwide, 30 sites in 20 different countries with the goal of studying 180 patients randomized to placebo or active oligonucleotide therapy to see whether this effect holds up in a large scale placebo controlled study. Now, there are issues with this. It's exciting to those of us in the field that it's being studied on such a large scale. Probably the largest yet randomized placebo-controlled study for Duchenne dystrophy, at least for a treatment that targets the genetic defect. There's the issue of safety. So the patients in this study do develop injection site reactions, nearly all of them. Most of them get proteinuria from as a result of the oligonucleotide treatment. There is still the issue of efficiency and stability of the treatment. It appears to be very long-lasting. The oligonucleotide lasts for at least a month after each injection. But the unanswered question is how much dystrophin is needed in the muscle to correct the clinical manifestations of this disease, to mitigate the clinical manifestations. And there's an interesting kind of situation that's evolving here in the realm of personalized medicine, in that there are many different exons that need to be targeted and multiple therapeutic agents that need to be developed. Every patient has a different mutation, and this treatment, the oligonucleotide therapy, has to target the specific exon that will be predicted to have a beneficial effect that needs to be skipped in each patient. So there's a long list of different oligonucleotides that need to be developed, each needing separate proof of safety and efficacy for regulatory approval. Now, we've had a lot of discussions with the FDA about how they might relax the standards if the first 2 or 3 exons skipped turn out to work out okay, if there's efficacy and safety or manageable safety issues. But still, it's a challenge here. So the first exon that's being targeted, actually by both companies, the Prosensa GSK study and the other company, AVI, is exon 51. That would be predicted to correct 18% of the deletions, 12% of the patients with Duchenne dystrophy. But then each successive exon, and 2 or 3 of these are now in earlier phases of development, would help, be predicted to help a smaller proportion of patients. So exon 45 would help 11% of deletion patients, 44, 8%, and so on. If you have 12 different therapeutic agents and can target them appropriately to the right patients, that would be predicted to help 3/4 of Duchenne patients. So we're on the beginning of a path here, that's taking us into a new area of individualized medicine, medicine that doesn't just target a disease, but targets, in some cases, a very small subpopulation of the disease. Now, yes. Now, what would clearly help here, something that's been shown, I think one of the models here is the neuroimmunology branch has developed MRI as a biomarker for multiple sclerosis that gives an early read as to whether an approach to treatment is being successful. There are limitations to how that's been applied, but it's been enormously successful in opening up multiple sclerosis for treatment. Our idea here is to develop a biomarker that would do the same thing for muscle disease. And what that needs is a non-invasive biomarker that will give an early indication of biological effect with exon skipping, so that we don't have to wait for a year or 2 to see if there's a clinical benefit. We don't have to do invasive tests like muscle biopsies to see if there's a biochemical benefit. What we're entering into now is an NIH imaging study, a study to be done here at the clinical center as part of a new phase 2 multi-center American trial of this GSK oligonucleotide targeting exon 51. We're working with Ami Mankodi and Karsten Bonneman in our group. We're working on this together with Andrew Arai and others at NHLBI to test outcome measures, cardiac and skeletal muscle MRI and ultrasound. Here's from a group at the University of Florida, an image of muscle MRI from a Duchenne patient and control. See that there are quite marked differences. The question that we're addressing is whether these differences start to change, start to show a structural response in the muscle, in the cardiac and skeletal muscle, before we can see clinical manifestations of benefit. And the protocol was just approved, and the first patients will be coming in the next few weeks. Well, I'd like to go on and talk about another disease, and the rest of the time we have available, another severe childhood onset neuromuscular disease. This is spinal muscular atrophy, autosomal recessive disease. Duchenne muscular dystrophy affects about 1 in 3,000 boys. This disease affects maybe 1 in 8 to 10,000 babies. It is, as an autosomal recessive disease, is probably the most common severe hereditary disease of infancy. The carrier frequency is remarkably high. So in European, Asian, North American populations, the carrier frequency is about 1 in 40. So in this room here, there are probably, at least 6 or 8 people who are carrying this mutation, would be at risk of having children or grandchildren affected if you had a baby with somebody else who was carrying the mutation. It is a disease that causes early onset progressive symmetrical muscle weakness. In its most common severe form, this disease presents early in infancy, and the babies are floppy, like rag dolls, as shown here in a picture from Victor Dubowitz's textbook some years ago. And the weakness is progressive, and it's associated with muscle atrophy. So this is a problem probably not primarily with the muscle so much as with the motor neurons that innervate the muscle. There is motor neuron loss and consequent denervation of the muscle that causes this weakness. The gene was identified for this disease back in the mid-90s, was given the name, a French group identified it, given the name survival motor neuron, or SMN gene. It encodes, the gene encodes the SMN protein. Yeah, I like this picture. This is a picture from the Dominican Republic, B. Bilikova here, and we saw a patient there with spinal muscular atrophy. This disease is pretty widely distributed through the Americas and Europe and Asia. Interestingly, it's not so common in Africa and people of African origin. But this is also to illustrate, well, not only that I, as an adult neurologist, can examine a baby, but also, I'm sweating a little bit here, but I think it helps to illustrate the disease is not always as severe as it was in the last slide. Some of the patients have later onset, are less severely affected. Some can have onset even and long-term survival into adulthood. So it's a disease which is most commonly pretty severe, but there, as with Duchenne and Becker dystrophy, there are milder variations. And the genetics is interesting. So this SMN gene is present in more than one copy. We have variable numbers of copies of the SMN gene, and it comes in two different flavors. There's the SMN1 gene, which is mutated in the disease, and another version of the gene called SMN2, which is nearly identical to SMN1, but differs at a single nucleotide that leads to a different splicing of the transcript. So this nucleotide difference is in exon 7, and it affects the splicing of exon 7 such that the transcript, the messenger RNA that comes from SMN1, is full length. SMN2, the majority of transcript is missing exon 7. And again, this leads to a protein which is unstable and rapidly degraded. SMN2 does encode some full-length transcript, and this is enough for patients to get by. A complete loss of SMN, at least in animals, is embryonic lethal. The patients with the disease, though, who have mutation of SMN1 still have SMN2, and they have reduced but not absent SMN. The SMN2 gene itself is present in multiple variable number of copies. The more copies of SMN2 that a patient has, the milder the disease manifestations. So the severely affected patients have no SMN1 and maybe 2 copies of SMN2. The more mildly affected patients, such as the one in the Dominican Republic and others with later onset, have 3 or 4 or more copies of SMN2. This leads to more mild disease manifestation and points to the likelihood that increasing, if we had a way to increase SMN levels, that could be an effective treatment for this disease. Now, what is SMN? We talked about what dystrophin is. SMN's normal function, well, at first it wasn't known. It was known that it is ubiquitously expressed. It's expressed in every cell, every nucleated cell of every organism, from humans all the way down to yeast. It's known now to play an important role in spliceosome assembly. It puts together the ribonuclear proteins that form this complex that's responsible for splicing all genes. And that's a clear function, and it probably has other functions as well. In particular, there's evidence to support a role in the axonal transport of messenger RNA. So it binds to proteins that bind to messenger RNA for cytoskeletal actin, for example, and that transport the messenger RNA from the nucleus down to the nerve terminals, the growth cones and the nerve terminals. Neurons may be particularly dependent, particularly motor neurons may be particularly dependent on having protein translation occurring at a distance from the cell body. And this is a great distance from the cell body in cellular terms. So why are motor neurons vulnerable to decreased SMN? Well, they have very long axons, multiple targets. The muscles that become weak early in SMA are innervated by motor neurons at a ratio of about 1 to 1,000, or at least up to several hundred. One motor neuron innervates many muscle fibers. Those nerve terminals on the muscle fibers are 1,000 times the size of nerve terminals in the brain. So motor neurons have an anatomy and physiology that may make them particularly vulnerable. They may be particularly vulnerable to messenger RNA splicing defects that come from abnormalities or defects in spliceosome assembly, or they may be particularly dependent on the axonal transport of messenger RNA. But the point is that correcting, whoops, yeah, I think that was it, yeah. Correcting the SMN deficiency is an approach to treating this disease. Now, since the gene was discovered, there are a number of different model systems that have been developed. First, cell-free biochemical assays, just splicing assays or ribonucleoprotein assembly assays, and cell culture assays. And then a series of animal models have been developed. SMN-deficient worms, flies, fish, and mice. And in each of these cases, there is a motor neuron degeneration that occurs. And they each offer different kinds of insights to the disease. Say the flies, when they outgrow their maternal supply of SMN, the larvae start to crawl along slower. They don't make it to adulthood. The fish, like our goldfish at home, kind of go belly up. and the mice have a phenotype which really looks a lot like the human disease. So mice, this has been known now for about 10 years that a couple of groups developed mouse models 10 years ago. This group in Taiwan and another at Ohio State University, Arthur Burgess, developed mice with SMN deficiency, not a complete lack, but a relative deficiency of SMN. And these mice are small and very weak, starting within the first few days of life, and they die. The patients with type 1 or the most severe form of SMA may die within a year or two. These mice die within about 2 weeks. Now, the group in Taiwan that developed these mice also tested treatments. It was interesting. I think when this first came out, it was secret for a while. There was a secret in the field as to what treatment was having an effect. The word leaked out from Taiwan that it was one of the first 20 agents that they tested in these mice. My brother, who's not in biomedical research, said, well, it probably began with A or B. And sure enough, it was butyrate. I don't know if they had to go further in the alphabet. But the sodium butyrate, when given to the pregnant mice, the mothers of the mice who were affected, would increase the survival of the offspring. Now, sodium butyrate is not a drug that's used in people, but it is representative, perhaps. It's not a very potent drug in this class, but what it is likely to work as a histone deacetylase inhibitor. Now, so this opened up the possibility of treatment of SMA with other more potent histone deacetylase or HDAC inhibitors. So gene expression is largely regulated. An important way to regulate gene expression is the acetylation of histones and other proteins in the nucleus of the cell. In general, when the histones are acetylated, it opens up the chromatin structure. It allows transcription factors to get into the genes and allows gene expression. So there are in the cell a number of different acetylases or acetyltransferases that put on acetyl groups, and correspondingly a number of deacetylases that remove acetyl groups, and the combination affects the levels of gene expression in the cell. Deacetylase inhibitors, which have been developed for hemoglobinopathies and cancer treatment as a way perhaps to work on tumor suppressors, these block the deacetylation of histones and increase gene expression, and they increase the expression of different genes to different levels, but they in particular increase the expression of SMN, the gene, the protein that's deficient in SMA, spinal muscular atrophy. So a group in our lab, actually this was first done by a medical student, a CRTP student who was in our lab, got this experiment to work like 2 out of 19 times, I think. and it's her first experience in the lab. But an excellent clinical fellow, Charlotte Sumner, now at Johns Hopkins on the faculty, got this experiment to work reproducibly, and it's been reproduced in other labs, a number of other labs elsewhere around the world now, that another HDAC inhibitor in common clinical use of valproic acid, used for psychiatric disorders and for seizure disorders, that valproic acid works as a HDAC inhibitor and increases SMN levels in fibroblasts from patients and in motor neuron-derived cells, mouse motor neuron-derived cells. It does this is with a reporter construct. It does this by activating the SMN promoter. Now, it takes pretty high levels of valproic acid to have this effect, millimolar effect, but these are the levels of valproic acid that are used therapeutically. So I should say a more potent and selective HDAC inhibitor, tricostatin A, was subsequently tested in our lab and shown to have a benefit in these SMA mice. It's work done by Amy Avila and Charlotte Sumner and others in our lab. It's a, well, you know, kind of a modest effect in increasing the survival from 15 up to average of 18 or 20 days. Here you see an untreated mouse and a treated mouse. This shows, in this case, the treatment was started not in treating the pregnant mother, but treating the mice after they started to become weak. So treatment after the onset of disease manifestations can be beneficial. And more recently, a group in our lab, Deborah Kwan and Barrington Burnett, have looked at using a clinically used agent, bortezomib, or Velcade, an inhibitor of proteasome pathway that decreases the degradation of SMN. So this combination of increasing the expression and blocking the degradation of SMN has a greater effect, a greater beneficial effect of increasing the survival. In both cases, a significant effect, more in combination than with tricostatin alone. But another interesting study, it was done by Heather Narver and people in our lab. Heather is a veterinarian, works over in Building 35. I think she kind of took, I don't know if she's here, kind of took pity on these mice who were so severely affected by the disease and gave them extra nutrition. So would carefully feed them formula together with the TSA. Now, nutrition alone did not have a benefit, but nutrition plus TSA led to a remarkable increase in the extension, in the lifespan, so up to 170% increase in survival. So, you know, I think that's a message to keep in mind, that nutrition together with treatment to increase SMN can enhance that effect. Now, based on these findings and others, several groups have entered into clinical trials now of drugs designed to increase SMN. A group in Italy used phenylbutyrate, another HDAC inhibitor, and a group, based a multi-center trial that was organized at the University of Utah, Kathy Svoboda, used carnitine and valproate. Carnitine to counteract the effects of valproic acid on mitochondria and liver function. Neither of these studies really showed a benefit, maybe mixed results with this one. But the sense is that these were not the kind of potent and specific HDAC inhibitors that are needed to increase SMN and have an impact on the disease. Now, what's become available over the years since this gene was discovered and its function's been elucidated is a number of different assays that can be used to screen for new drugs and develop compounds to approach this disease clinically. You know, assays based on the promoter, on the retention of exon 7 in the SMN2 gene, assays that measure the protein levels, so they can pick up agents that might block the degradation of the protein as well as enhancing its expression, or assays that look at the function, the downstream function of SMN in RNA protein, ribonuclear protein assembly, or that increase the survival of cells that are deficient in SMN. And there are now a number of drugs that are different stages of development, you know, based on these assays. So first, a study in Europe with a neuroprotective agent developed by Trophos. Families of SMA, a voluntary group, has helped fund drug screening efforts and drug testing efforts, pass these compounds on, quinazoline to a company, Repligen, and tetracycline derivatives are being developed by Paratech. This Trophos compounds in clinical trials in Europe. These are heading, the quinazoline's heading towards clinical trials by Repligen. And then here at the NIH, the NCGC, the NIH Chemical Genomics Center, working with Elliot Androphy from University of Massachusetts, has done a drug screen. It was just published last month, a drug screen and optimization of the compounds that work on a reporter construct for SMN, and these are aryl piperidines. We're going to be testing further in our own lab another set of compounds that carry potential as treatment, as small molecule treatment for this disease. But meanwhile, there are some very interesting developments just in the last year or two with biological approaches to therapy for spinal muscular atrophy. SMN gene replacement, gene therapy, and again, oligonucleotide-induced, not exon skipping, but exon retention. Oligonucleotides that promote the retention of exon 7 in the transcript from the SMN2 gene. Now, first with gene replacement, remarkably good results, I think, with injection, again with AAV here and lentivirus in mice and in muscle. You know, injecting genes to deliver into motor neurons in the spinal cord, nice levels of expression, particularly when the systemic delivery is given early in the first few days of life, and nice results here in monkeys even later. Now, last year there were 3 reports of remarkable benefit with early postnatal delivery of SMN with AAV vectors. Here's one from Brian Kaspar's group at Ohio State, published in Nature Biotech. Here you can see that first the gene is delivered into the motor neurons in the spinal cord. You get a marked increase in SMN levels in the spinal cord and a nice behavioral benefit in terms of writing time, how long it takes these mice to roll over. Here the SMN treated mouse, here a control untreated mouse and basically a placebo treated mouse and a control mouse. And look what it did to the survival. So here's the placebo group where they die in 15 days, and here's the SMN treated mice that survive up to 250 days. So boy, this is really having a dramatic effect, basically curing the disease with a single injection in the first few days of life. Now, these mice die pretty quickly. If you wait even until 10 days after 5 days of treatment, this effect you don't see it. But if you give the treatment day 1 up to about day 5, you can cure the disease by replacing SMN. So the results of this plus the HDAC experiments indicate that there's a window of opportunity here in the very earliest stages of the disease where if you correct the defect, you can have a pronounced effect. This is with intravenous delivery. Now, oligonucleotides, here the work has been done primarily by Adrian Krainer's group up at Cold Spring Harbor, working with a company, ISIS, out in California, have first screened a whole series of oligonucleotides that match sequences on either side of exon 7. The goal here, again, is to promote the inclusion of this exon so you get full-length transcript from the backup SMN2 gene. And what they find is when they inject this oligonucleotide, just single injection into the cerebral ventricles of these mice, you'll get long-term survival and expression inclusion of the exon for up to 6 months. So one injection, 6 month effect. Now, what was just published, I'll show on the next slide, just published last week in Nature is another study from this group that shows that you don't have to inject it into the cerebral ventricles. You can inject these oligonucleotides subcutaneously, as in the Duchenne trial, and you get a remarkable survival benefit with subcutaneous injection of this oligonucleotide. And remarkably, it's working on increasing SMN, but not in the motor neurons. It's working, it appears to be working by increasing the levels of SMN in the liver that leads to increased production of a deficient insulin-like growth factor binding protein that leads to IGF-1 deficiency in these animals. So interestingly, systemic correction works not directly on the motor neuron, not by getting into the central nervous system, but by working outside the nervous system in the liver and correcting the defect. So here's the survival benefit that they get. Again, the untreated mice dying within 15 days, normal mice survival here up to 500 days, and with increasing doses, subcutaneous injecting just in the first 3 days of life, they get survival up to, up to 500 days. So they're effectively curing this disease with just 2 injections in the first 3 days of life. Now this shows, this is on the handouts that you got in the poster, the effect at the neuromuscular junction. The untreated mice have, here the axons going into the motor end plates, and the untreated mice have a kind of retraction of axons from the motor end plates, which is corrected by the oligonucleotide therapy. Here's what happens to the IGF-1 levels in these mice. So this hadn't been noted before, but without treatment, these mice have 0 IGF-1 in their circulation. IGF-1, as I said before, is a trophic factor for muscle, promotes muscle regeneration. It's also known to have atrophic effects on motor neurons. So the lack of IGF-1 in these mice is likely contributing to the motor neuron degeneration. The dependence, there may be a critical dependence on IGF-1 in this critical early stage of the disease in the first few days of life. And this is not due to a problem with IGF-1 itself, but to a deficiency of IGF-1 binding protein, a deficiency of the binding protein which is corrected by the treatment. A really remarkable study. Now, in closing, I'd just like to mention another interesting observation from Petra Kaufman, who is at Columbia University, recently recruited to the extramural clinical trials group at NINDS. She and her colleagues a few years ago did an interesting study of the natural history of the severe type 1 form of spinal muscular atrophy, also known as Werdnig-Hoffmann disease. And they found that patients who were diagnosed in the 1980s and through the early 90s had a very limited survival. So most of these patients died within 1 year of age. A few had long-term survival. But when you look at patients who are diagnosed later, in the late 90s through the early part of this decade, of this century, the survival's been much better. Now, if we'd seen a response like this with gene therapy, oligonucleotide therapy, small molecule therapy, we'd be celebrating. But this is without any kind of treatment other than optimizing standard care, particularly respiratory nutritional support. So it shows first that the natural history of this disease is changing. The same thing's true, I should say, for Duchenne muscular dystrophy and other neuromuscular diseases. And it's probably because of optimizing existing care. So there's a real role for doing that. It points, the analogy here, or a model for us here, is what's happened with cystic fibrosis, a non-neurologic disease, of course, but one that has shown a remarkable improvement in survival from the 1940s and 50s, where the patients survived less than 10 years on average, to today, where survival is up to age 40 or better. And there have been other treatments that have become available for cystic fibrosis, but it's primarily a more and more aggressive optimization of existing care with respiratory and nutritional support. A lot of what's helped to make this happen is what the Cystic Fibrosis Foundation and the clinical centers have done to make morbidity and mortality data openly available for comparison and optimization between centers. And I'd argue that that kind of approach, which has also been applied to cardiothoracic surgery, is something that could help patients with neuromuscular diseases and other severe diseases. We just have to apply this kind of approach. It's easy to say. So the goal here is to be able to have a treatment that doesn't work just here in Bethesda or at major academic medical centers in the United States, but we could bring back to the Dominican Republic, Los Frios, right, and places like this, to help patients with this disease wherever they are. And I think that's an achievable goal. We can almost reach it. I'd like to just close with this quote from Reinhold Niebuhr, a philosopher and theologian from the last century, who wrote, "Nothing we do, however virtuous, can be accomplished alone." Clearly, this is a group endeavor, and it's something that takes a lot of collaborations outside of our group and within our group. This is our group from our outing last spring. Not everybody in this picture works in the lab yet, but this shows some of the people who work on the Duchenne muscular dystrophy trial. Amiman Cody, who's here in the audience, and Elise Schindler, Angela Kakinis, and the people working on spinal muscular atrophy, Barrington Burnett is an important member of the group, but he's not in the picture. He was picking up his child at daycare. But he's back there by the microphone. And Deborah Kwan, Catherine Braccino, Modibo Sangare, who's looking at why the carrier frequency, or confirming and looking at the carrier frequency in West Africa. So thank you. Sorry, went over a little.
We could take a couple of questions, but I remind you that there is a reception right after the lecture across in the library. So if anybody has a question, please go to one of the microphones.
Well, looks like after a lot of frustrations, the antisense are making some headway. So what is the length of these oligonucleotides, and what are the specificities and the degradation rate after administration?
Yeah, the oligos, I don't know specifically, but I think they're in the range of 16 to 20 nucleotides, and they're degraded very slowly. It's actually frighteningly slow. The half-life is, in the Duchenne trials, is about a month. And, you know, the problem with that, if you get into toxicity, is it takes a long time to clear the agent out. The advantage, on the other hand, is that it, you know, a single injection can give you, you know, months of response. In the mice, it's a lifelong response to a single injection.
So at least for the clinical studies, you will be doing the pharmacodynamics of the degradation and looking for the MRI. Well,
yeah,
wish you good luck. Thank you.
Oh, thanks. Yeah, and you know, I wouldn't give up on the small molecules. This is the way therapeutics development is. It takes perseverance. There's a lot of back and forth, and the goal is to learn something with each trial that helps to better inform the next one. And I think, you know, some combination of therapy may be the answer. Oh, yeah.
Hey, Kurt, that was just a great talk, wonderful overview. I had a question about SMA and your points about the need for early recognition and treatment. So do you have any remarks about the possibility of newborn screening? And, and then, of course, in the developing world, that's an even larger potential problem. Yeah,
we've had a lot of talk about this, you know, to have more widespread carrier testing and newborn screening. You know, it's interesting. We've had some interesting meetings about that. With carrier detection, you know, people are arguing both sides. If we put too much into carrier detection, what about, you know, developing a cure for the kids who are already affected? The voluntary groups are on both sides of that issue. The geneticists and the obstetricians are on different sides of that issue. I think what's, you know, where the conundrum is, you know, there'd be a good argument for widespread screening once we have an effective treatment that is shown to only work early. Right now we have a lot of evidence that a treatment needs early diagnosis, but it hasn't been proven yet. And once we have, then I think everybody will go along with the idea. But there's a lot of work in that direction. NICHD is putting a lot into assay development, making cheaper assays more widely available for this disease and others. And I think the future is going to have this be incorporated into newborn screening panels. It's already being you know, widely done in places like Israel, for example. I think it'll happen in the United States too. But it's in an interesting stage right now where you need to show that it's necessary and effective to make early diagnosis in a situation where it's pretty likely to be the case, but it hasn't been proven yet. Yeah.
Do these two types of dystrophy affect extraocular muscles?
I'm sorry, could you say that again?
Do they affect extraocular muscles?
Oh, extraocular. Oh, yeah. So you really don't get, it's interesting, you don't get much eye muscle involvement with Duchenne or Becker dystrophy. Geez, you know, I've seen, you know, some of my patients get autopsied and the muscles are just completely gone elsewhere, but you still have extraocular muscles. Likewise, spinal muscular atrophy, you don't have much denervation or involvement of the extraocular muscles. There are other kinds of muscular dystrophy that do. Ami Mankodi has worked on oculopharyngeal dystrophy, a repeat expansion disease that affects the eye muscles in particular. And then, you know, we see mitochondrial disorders that affect the eye muscles preponderantly.
But so this resilience of eye muscle cannot give you some idea on possible therapies?
Yeah, you know, it's an interesting question. I think, you know, so I said that the mice are less severely affected than patients. Dogs are more severely affected like patients with Duchenne dystrophy. I think it might just be a matter of muscle size, it's a structural protein that big muscle fibers and big muscles have more of a dependence on this structure, just like a tall building would have more dependence on structure than a short building. But that's kind of a hand-waving explanation. It's just one possibility. It may be the innervation ratio, other aspects of the muscle that make the difference, and maybe there's something to be learned by that. John Porter is now an extramural NINDS, did work on that when he was at Case Western Reserve before he came here, and he's still very much interested in that question. Yeah. That's not the John Porter our building's named after, by the way.
Okay, thank you for a fabulous lecture, a great Astute clinician lecture.
Thank you.
Please join us at the reception right across the hall.